So let me welcome everyone to the theoretical computational biophysics seminar. As I was telling our guests, it's so good to see people in the tower room again more regularly. So I'm very pleased to introduce Dr. Jin Yu, one of us, actually a former group member from theoretical computational biophysics. Maybe I use one of the microphones.
I take one.
Yeah, there. Actually, we can. No.
I think you need to switch it on. So
The red one?
the white one.
This one. Okay, good. Yes, so it's a pleasure to introduce Dr. Jin Yu from Department of Physics and Astronomy, University of California, Irvine. As I pointed out, Jin is one of us, actually a former group member from Theoretical Computational Biophysics group, and she kindly agreed to visit us and give us a seminar. So by way of introduction, Jin was trained as a physicist in the famous Tsinghua University in Beijing. She received her BS and Masters of Science degrees over there, and then she moved to UIUC for her PhD with Klaus Schulten. So she did some simulation already on DNA protein system, collaborating with the group of Tegi Pa in the physics department, and really making nice prediction of how the mechanics of these very complex systems work and how these molecular machines actually can work. And then after that, she moves to Berkeley to work with the group of George Oster, the late Oster group, a very well-known group in mathematical modeling of molecular machines, where she actually got involved in physical and mathematical modeling of the same molecular systems. So I guess the focus of her work continued to be on the way proteins and DNA molecules interact and how DNA can guide the motion of proteins and so on and so forth. In 2010-11, actually, she moves back to China in Beijing and joins a research institute there, focusing on computational sciences and their application in biomolecular system. She actually develops a nice group there already, but then fortunately we got her back in 2019, if I'm not mistaken. So she got recruited to University of California, Irvine, Department of Physics, with joint appointments in the Department of Chemistry, as well as in one of the Simons research institutes that they have on campus. So it's a great, I mean, Jen has again continued her work. So you're going to see the work that she has done on the DNA protein theme. And just so you know, she was one of the new and notable lecturers at the last Biophysical Society meetings in San Francisco, in case you missed that. Thank you very much, Jin, for coming all the way from the other side of the country to visit us. I hope that you see changes around the campus, and we look forward to a seminar. Please.
Okay. Thank you so much. Thank you, Emad, for inviting me. And it's my great pleasure and my honor to be back here. And okay, I'm going to share with you my research in these years. So I'm going to put the focus on the protein search on DNA, and then about transcription, RNA polymerase, and then a little bit about DNA mechanic feedback and supercoiling. So basically 3 parts, but I'll focus on first 2 parts.
Mm-hmm.
Okay. So as you heard, I were one of you here. I come from here before that. So I were from Tsinghua University, so where it has the same kind of dome building there. And then, so in this big group, Klaus was here, Emad is here, and I were there. Okay, so it's a large group. So yeah, Oh yeah, you will see the, yeah. Right? Anyone you want to point out? Okay. Yeah, so mainly I work with the protein DNA system. Oh, but I do have one membrane project, actually work with MR. And actually that one is my most cited paper as the first author up to now. Okay, so I work with Klaus and collaborating with Tekipa mainly on helicase, holiday junction, this kind of stuff. So this is the title of my thesis. So I do MD simulation primarily, but then I want to approach something else beyond. Okay. So actually follow the same kind of theme. I study many the protein machinery dealing with DNA, and particular we're interested about that in the central dogma. So those machines expressed, but some of the machines play metabolism, some play structural protein, and some of them come back to regulate the central dogma. So these are the things we're most interested. So I kind of also influenced by my postdoc advisor, George Oster, and collaborative lab, Carlos Bustamante lab, who doing single molecule physics, many about machinery in central dogma. Okay. So now the system I'm gonna focus is first, it is this transcription factor search on DNA. It's not a new topic, but I sort of revisit this topic with our technology, which you are familiar, but we take some chance there to see if we can, what we can contribute. So transcription factor, basically nothing strange, they just do diffusion with thermal fluctuations, no energy resource. But in the end, they can find some place on the genome, so these specific sequences, so then they contribute to the function. So this is amazing thing, how they search, okay? Then for the RNA polymerase, which do transcription from DNA to RNA, this is a machinery work from some bacterial phage. So basically it's a model system. We'll see this machinery. Now the difference is, this is molecular motor. It consumes here actually the nucleotide, ATP, GTP, whatever, which the components want to be added to the synthesizing chain. So the big difference here is this is purely by diffusion, but here you have free energy resource. But there's something connected. So this is also informational wise. You want the fidelity control. So we're concerned about that. The mechanic thing, the chemistry thing, and the information. In the end, I talk a little bit about the protein itself, because the protein is not purely passive here. By forming supercoiling, it gives feedback to the system. So that will be a small, the last part of briefly talk about. Okay, so these work minutes, I started from the center where I worked in Beijing and continue later on. So these are my former group and the funding and computational resource there. Okay, so first about this protein search on the genome. So it's not a new question. So people realize it's an important issue seriously, starting from 1970. When they find actually lac repressor, they can seamlessly bind to the target sequence even at the rate higher than the diffusion limited. So they ask a question why they can do that so fast, and even among this genome space, how can they locate this a tiny amount of information there? Okay, so in brief, the research we conducted right now is just at a very local region of DNA. So you are familiar with this, use atomic simulation to locate the movement of that. I'll explain why this is important because it seems straightforward, right? They should move, you should detect that, but why there's some significance there? And we actually kind of can get a rotation coupled motion of this small transcription factor protein. We can also see stochastic variation. Basically, we just see a one base pair forward, backward there. And we kind of can deduce the energetic ruggedness about 1 to 2 kBT with consistent with previous measurements. And we locate actually the collective hydrogen bond dynamic at protein DNA interface. This is the one thing to be notable. And also we watch protein reorientation frequently on the DNA. And we also find that there's a biased DNA strand, this blue one, the protein favor. So these things I'll gonna explain in the following. Okay, so
sorry,
yeah, please.
So this chromosome that you showed, what fraction of that is like a free piece of DNA that these proteins can bind? Like this part that you're showing at the bottom.
Yeah, but you know the thing is this is put in a eukaryote system. It's more complicated because they do have this folded structure, and then you have open frame that is many protein can bind, taking the chance. So I think the opening and closing of this nucleus kind of impact on how transcription factor bind. But I think in search-wise, initially when people focus on this search, they start from bacterial cell. So in that sense, I mean, you do not have complicated genome structure, but you do have this DNA exposed there, you want to search.
Right.
But definitely this search problem in the eukaryote system will be more complicated.
So is it fair to say that in our body, you only work with these open parts of the DNA?
Yeah, that's the current understanding. So the regulation then come of this chromatin open or closed. So it's something matters for the regulation.
Yeah. Cool.
Okay, so from this bacterial cell, a kind of a theory built actually in 1980s. So people propose this facilitated diffusion model that is protein alternated between 3D search and 1D search. So they do this sliding. Sometimes they also do some variation like jumping or intersegment transfer. So overall, they try to say, okay, if you want to locate some specific sequence here, but you have sliding, it's like you amplify this region. So you give amplified signal. So that help to find the location specifically. And also by some quantitative model, they just suggest about half the time they spend on 3D, about half the time on 1D. That could be a near optimal search strategy. Of course, that was developed from bacterial cell. It doesn't necessarily apply to higher organism, but we'll get some idea there. So actually, for the detection of diffusion coefficient on DNA, it also started early, and people deduce something and estimate some upper bound. So following up, there are measurements. So for example, RNA polymerase, when it's not doing elongation yet, before the recognition open the bubble, it's still sort of like a transcription factor. So it's also have this diffusion search rates measured, and also for some transcription factor, this much of diffusion coefficient measured. Okay, now we just want to do some unit conversion to remind you. So the upper bound at order of magnitude, when you change the unit, it is giving about 1 base pair per microsecond. But this is about upper range. So I think people sit here realize very clearly we can do microsecond simulation. So it is this checking of the literature and thinking about give us some initial confidence. If we try to do MD simulation, we should be able to see something there. But meanwhile, you can see for experiments, if you want to detect the base pair movement, you want to have the time resolution microsecond or at least below millisecond. And then you have a spatial resolution, several astron below 1 nanometer. So just dealing with one high resolution, high spatial or high temporal is achievable. But to dealing with two simultaneously, actually it's challenging. So people, I think nowadays still struggling with achieving this resolution simultaneously. So this gives us the chance, if we somehow simulate this in empty simulation, we can show something which have not been exactly measured experimentally. So this is our motivation, and also we want to take a little bit risk and chance to if we can do that. So this is pretty much the motivation we want to. Actually, RNA polymerase, when the molecular motor or on the other helicase, actually I believe the mechanic movement is similar, the rate, but the thing happen is different for molecular motor. So they are dominated by chemical cycle. Each cycle usually happen in a millisecond time frame, but that's what happens slowly there. It is usually chemical reaction or some conformational rearrangement. So that makes those kind of molecular motor different and also maybe easier to measure. But when people talk about like stapling size, protein DNA interaction, I believe what we find in the transcription factor also matter for those like helicase, polymerase, because they basically just protein move on DNA. Yeah.
So if you have projects in the group that, let's say, they're looking at how a piece of nucleic acid is moving inside the protein, which is the reverse of the process you have described. So we should expect one base pair per microsecond kind of.
This is upper bound. But also be careful. So this is a double strand DNA. For example, if you're dealing with helicase, this is single strand. So attachment could be stronger. So time scale may be different. So
right.
Yeah. So this is upper. So it could be slower.
Fair enough. Got it. Okay. Thanks.
Okay, so with this, we have this belief, but then we have initial struggle. We simulate, we have collaborator with specific protein DNA binding complex. We start there, we simulate specific binding. Sure, you won't see movement because it's specific binding. And then we simulate a lot specific binding. We couldn't find that. We change, like we want to change the periodic boundary condition. We're thinking, should we make the DNA infinitely long, or should we constrain DNA somehow? I mean, we didn't succeed either. But meanwhile, we actually spot some very close interaction between DNA with hydrogen bonding. So the thing is, in this group, people know this is natural thing, right? But for some other people, I mean, know some molecular biology, but not into detail. They may think that the protein DNA, when they do specific recognition, they form hydrogen bond. But they may think hydrogen bond won't be formed between backbone during movement, but it is not. During the non-specific binding or movement, there are always hydrogen bonding formed at the backbone. So we realized maybe our DNA was stuck with hydrogen bonding. So then I try to think what we can do. So we guess probably we use homogeneous sequence, poly-A. And then the student tried again. And after 2 or 3 rounds, futile, and then we really catch the motion. So that's I'm gonna show you. So these are the so this is one base pair. This called forward movement.
Okay, I see.
So it looks simple, right? But it's sort of the first time you see that because experimentally you cannot see that because it's too fast or too little. So yeah, we played the game. So you see that. So if we check, so this overall it is a 10 microsecond simulation, and you can see that actually the first half is pretty much stuck there. So we have to some belief it will happen. We also didn't do accelerated thing. So you have experience, you know, it's good, you do perturbation. But on the other hand, I mean, the system is very sensitive. If you do something, you have the risk to disturb. So because we know the time scale, they can upper bound, we have some knowledge from the experimental collaborators. They have some single molecular estimation of diffusion coefficients. We sort of know this is a fast one, and this is small. So we kind of put the belief. So we finally find that. So it's I mean, after several months, we almost still want to give up. Nothing show, right? Why you believe that? I think. So here, nothing strategy. Everybody can run the simulation, but I guess at that time it's just a little bit belief support us with this. But we have to estimate the quantitatively. Is that realistic or it's not? Also, we find if we put some force on DNA, actually it's not very good. Actually, the proteins start kind of doing things differently. We do not systematically evaluate, but I think that protein is sensitive to the DNA configuration somehow. Okay, then this is one trajectory. Then we sampled another one. So this time it's backward. And now see, it's not, so if you say this one is rotation coupled, so it is really track the groove. But if you want to talk about this as a story, I think you put your conclusion too early. So this definitely is not that regular. So it's just climb this this strand and cross from major groove move to minor groove. The same condition. We pretty much run 5 or 6 this 10 microsecond simulation. We spot 2, and later on we change force field. We spot another one, but you know there are several ones not spotted with anything significant yet. Okay, so then what we learn here from this backward. So initially, still kind of want to move forward actually, and then suddenly it just crossed this thing. Okay, so we sort of learned this. Stochasticity there, and actually we heard some informal communication, some experimental people. They said they sometimes find some fast motion. So we were just speculating. It's possible if they have large percentile of this type of motion, that will appear much faster, right? Once you cross this, immediately you cross many base pair. But here you just the one base pair. Please.
Quick question: is this the initial structure? Is it the native structure for this transcription factor?
So no, actually, so what we got from crystal structure is the specific binding complex of that. So specific binding complex in DNA, then we first construct a non-specific one, just mutate the DNA part, and then we also change that. So actually, there is risk that you probably will have huge conformational change, but you do from a specific one to the non-specific one, you just do not catch. But we try to think it's unlikely because first, this is a pretty small protein. Basically, it doesn't have much there, right? So the second is later on when we detect the free energy of this, it matches experimentally. So it's like a 1 to 2 kBT because if you are, so there are actual theory saying about, they suggest the protein search should have 2 modes. One is search mode. The other is recognition mode. That means they have 2 conformation. So the conversion between them could in between microsecond to millisecond. So that gives us this risk that it could be something as you asked exactly. So may not detect that still have this possibility. So but here it doesn't look like because it doesn't have much structure, and if this is really in the recognition mode, the free energetic will be very high, but we detect pretty low. So these give us some kind of evidence to intend to think it's not, but I would not put it too general. I would say if you have a bigger protein, so I think they could exist two mode, two conformation. So lac repressor, for example, I think it's possible actually. So I always try to talk the scenario I talked today. I mean, as just for domain protein, you can see it can fit into groove, right? So for some bigger protein, it cannot fit. I think the scenario may not be the same.
Right. So
Okay.
because of the mutation, you want to simulate the search.
Oh yeah, we
right?
just want to, yeah,
So
exactly.
you want to see whether it kind of comes out. Or
Yeah,
whether
yeah,
it
because
follows.
specific is they're supposed to stay there.
Yeah.
Stay there, not, yeah.
Good.
Yeah, so that's we just technical, but there is some risk you doing that, especially if you have a giant one. So you may convert your DNA, but the protein may stay in the same configuration. So yeah, please.
I mean, based on this stochasticity, there shouldn't be any directionality of this movement
exactly.
in biology,
You're
right?
right. You're right. So that's we to take a forward backward. Molecular motor is not. They should the majority. They should go to one direction.
Sure.
Even you can still detect some backward, but in the probability wise, they have dominant direction.
Okay.
Helicase or polymerase, they all like that.
Right.
Yeah.
Interesting.
Okay. Okay, so then I want to show you that it is really going one base pair. The criterion is not actually according to the center of mass. Actually, if you measure center of mass, you can even see that the specific binding complex have a lot of fluctuation of center of mass. Initially, my students asked me, "Oh, is that moving?" I said, "Oh, do you think it can moving because the hydrogen bonds all stick there? How they can move, right?" So now we have to show the evidence. So you see that this is initial configuration. This is a schematic of hydrogen bonding. So initially you have this configuration for a long time, 5 microseconds. They just have this front residue probe to this one. But other than that, it's still like this way. But something happens soon. Suddenly it's broken. And then you can see just barely two maintained.
Yeah.
Okay, then continue. So this is the key one staying there. So if we come back, going this way, you see then they recover a bit, and then till this. Now you see at the end of this cycle about 7 microseconds. You see what? So all the hydrogen bonds shift one base pair. So this really gives us confidence. Okay, this, I think it is that moved. I think this poly A give us advantage, like they are more synchronized, I feel like. If you have heterogeneous the DNA sequences, some hydrogen bond may be very sticky. So they will eventually broken and reformed, but maybe take longer time. So that is, I guess, our bet on the homogeneous sequence, maybe is a good choice. Yes. So okay, so this is a one base stapling. Okay, then we at that time we just do microsecond simulation. We do not do many number yet. So then we accumulate this much. We can. So this is basically when we have this forward movement. So this is rotation couple. When you cross the strand, then you have this to another diagonal line. Seems so. This is in the protein translational degree. This is in the rotational sort of helical thing. So mapped there. So free energy wise, you see that the dominant. I mean the amplitude will from 8 to 12 about, but the sort of fluctuation or standard deviation is about 1 to 2 kBt. So yeah, we also do vary the force field actually due to the request of a reviewer. Yeah, so we also captured similar. So actually, this is quite consistent with I think 2009, right? So Sunny Shea group, they measured this different transcription factor. This have this roughness of this diffusional. It's sort of in the search mode. So this is also evidence. Even we do not change, we use the initial configuration. We change it to poly A. So they give this pretty shallow phase. If it's really in search mode, it should be very sticky. So this is also evidence. We somehow think it's this is very simple protein. Okay, so later on we want to do more extensive sampling. So because the kinetics, we just sample a small number of trajectories, maybe we cannot get the statistics. So that we run extensive simulation and we try to construct a Markov state model from that. So people here, some people run that, you know this technique. So it's pretty standard here. Okay, then what we find here is, so we have a kind of three-state model. So what happened fast is this protein fluctuation, central mass. If you check the central mass, you will see that no matter which system, protein central mass always vibrating. So that gives the clue if some people do single molecule experiments. If they tag that on protein, you see the fluctuation is not necessarily they are moving. But the snow state happened here. It's a collective hydrogen bond dynamic. So they can have collective behavior. So all of them move, then you can go back. So this is a kind of story, but of course we take advantage. We already have an initial pass, because when do this construction, usually people want to a lot of time passed. Okay, so so people want to use some induced pass, but I think we here at, if you sample the pass you like, and then you do extensive sampling, then you get statistics. Okay, so then for specific and non-specific complex. So you see that you see protein central mass still vibrating there, but they kind of do nothing. But if you detect the actually, you see a lot of orientational change happen between the specific and non-specific system. But conformation-wise, if you detect, they almost do not change. So I'm just saying right now, it doesn't look like the search. They support this search, but it doesn't say it won't happen. It just want to say in some system, this theory may not work. Okay, so yeah, then this is I just want to show the hydrogen bonding here and specific protein DNA context. So you see actually here, this hydrogen bond formed more on one strand, but not that much in the other strand. So in a non-specific complex, so just change two nucleotides. So they have this trending, but less significant. Just one, prefer one strand. Then we just do protein DNA interaction in specific, non-specific movement forward or backward. We see that preferred DNA usually have lower this energy, electrostatic. Okay, and also it is most significant is specific, but especially when it moves, they kind of overlapping energy. Please.
So when during your simulations, do you observe the protein kind of unbinding from the DNA and then?
Not yet, but in coarse-grained simulation, I'll show you, because the time scale yet. So the fastest motion is actually diffusion motion. Actually, about 100, 200 base pair on average, they may dissociate. That is roughly estimated experimentally. So unbinding dissociation is a rare event.
I see.
Okay.
I was just curious if in case you do see reassociation with the same interface.
Yeah, but when we do coarse-grained simulation, you see that next. Okay, so let me summarize this atomic simulation part. So basically, we put the energetic thing, electrostatic, preferred strand. So you see always the trend. Then we have different systems, specific, non-specific movement, or then different force fields. So they always have trend. As you can see, the bias is most significant when binds to specific. So we are thinking probably this preferred strand help the recognition on the particular hydrogen bond forms also with space. This may help. So of course, I still think for small domain protein, this matters more because they really can bind tightly to the groove. The chance is binding to the groove give the hydrogen bonding chance.
Nice.
Okay, so bigger, especially for transcription factor, they could be dimer or many of them aggregate. I think the story will be more complicated because you need other coordination. Yeah. Okay, so this, oh, by the way, reorientation, I actually think is important. So basically, this protein doesn't have that much conformation to change, right? So, but it does reorient. So because hydrogen bonding is very dependent on your orientation. So I think this tuning of orientation on DNA is very important. Okay, so then you know that something we just cannot sample. Then we do coarse-grained model. So, so first we actually use some existing software and some model of DNA already developed quite a while. But I mean, the good thing is they can reproduce somehow persistence of DNA. They can also get some sequence effect, although hydrogen bonding definitely not there anymore. So roughly, they can have 1 order of magnitude improvement of efficiency, but they actually make the things tend to happen even faster because there's no hydrogen bonding. Basically, it's electrostatic.
Right.
Okay, so here just play the movie. So this is a regular ionic concentration. You see, dominantly is rotation coupled, but sometimes they have variation, right? Okay, then this is sorry. I want to. Okay, what happened? Okay, I just want to play the movie. They just get confused. I should try. You're gonna see that. Huh. So sorry for that. So basically, this if you lower your concentration, they will most of the time just tracking along DNA. So they do not do much because the lower the ionic concentration, the screening between protein and DNA reduced. So they are acting more tightly. I can play that afterwards. Yeah. I promise you. Okay, so but when you increase the ionic concentration, it's opposite. So the screening is very strong. Protein DNA interaction is very is loose, so they can dissociate from time to time. So you can see they move around. They can, I think, if you keep doing that, they can do hopping a lot. So that means if you can changing environment, the protein can behave in a quite a range. So stochastic motion are there. So do not try to present too much kind of story because stochasticity fluctuation are so dominant in these systems. Okay, so then with this, we can actually doing some statistics here. Just show the different ionic concentration. They kind of so this length scale increased. They just circle around DNA with larger range, and this just help us to do some statistic. You can check which fraction are more regular, which fraction a lot. So building sort of a computational assay on that. And also very interesting thing is even in coarse-grained, we try to detect if they have some base, like each time how long they go. So if we have this homogeneous sequence, majority is 1 base pair. But if we tune that to ATTA, like 2 base pair as a pattern, so you see there is more percentile of 2 base pair. So I talk about this because I know work with those like Tekiba or some other people working with stable size, so they matter a lot. So they think, why this variation? I mean, when I were in Bustamante group, they keep detecting like 2.5 base pair, whatever, kind of strange thing. So I think it's protein DNA interaction. So protein is very sensitive to the pattern on that. So of course here actually hydrogen bonding, the most informative part is not there. But I'm just trying to say, as long as interaction, even electrostatic interaction, it also gives the hint. I think in reality, if we really have hydrogen bonding, if you really run very long, I think they really can detect the thing. So here is just a hint. Okay, so then this is dimerical. We also run this coarse-grained simulation. So we actually see this inchworm. So PCR helicase, we have this inchworm, like you open, close, open, close, right? And there's a hand overhand. I think in this department of physics, they have, I mean, they have measurement of kind of myosin, this kind of stuff. So this kind of thing can be detected. Use coarse-grained. I think it will be pretty much robust. So we can also reverse actually coarse-grained to atomic. So this just this is not very, so this is not very refined, but you see some kind of events we can detect from coarse-grained simulation. Yeah, this is an advantage, although it may not necessarily be accurate. So we have to wait to see experimentally if they have yeah, but this gives some hint. So here with this kind of assay, we can actually detect different conformation, some waiting time, just like experimentally. So I think that's what you call it assay. So it's just help the statistic. Of course, the model is lost some information there. Okay, so meanwhile, we are doing something beyond, because as you mentioned, this dissociation, right? Dissociation, even in this coarse-grained model, is not quite much detected because dissociation happened like milliseconds or above. So even for coarse-grained, it's hard to develop. But then we developed this toy model. So it's just simple. We have electrostatic hydrogen bonds. This is not actually quite necessary, but basically this term. So our purpose actually to linking the binding, the free energetic, and the diffusion. I think actually when people doing kind of simple physical modeling or mathematical modeling have some confusion. They usually throw some energetic, but what is energetic? Energetic is here. So so actually, energetic is we have this, we map to UZ, this is a translational, and this vertical direction. You have this sort of potential mean force you can map there, but if you go diffusion, it is along this way, right, horizontally. So you have diffusion energetic. But if you do binding, it is reaction coordinate. It's vertical, but you can measure unbinding here, then you can measure unbinding here, and then you can unbinding here, and then you can compare them. This will give you the binding profile. So actually, it is what we are thinking. So if you do this, we actually can find their difference. So this diffusion energetic is the dark one, and the red is the binding free energy profiling. So this matters because actually, so for diffusion, actually the reorientation is sort of restricted because they are attached by hydrogen bonding. And so they are giving steeper energy profile. But when they do orientation change, they dissociate, they can have smoother one. I think this is time dependent. If you do real-time measurements at single molecule level, you probably will detect the diffusion energetic. But if you do like the bioinformatics, do some chipset profiling, what you measure would be this dark one. But this is under development. We still want to develop well and convince other people. We want to have some more extensive, maybe connection with bioinformatics level. Okay, so but we really want to push some physical modeling. I also want to do some machine learning stuff, taking advantage, but I think the physical modeling is sort of my motivation. But we can do it atomically and also do it physical coarse-grained level. Okay, so so this part I acknowledge that. So maybe okay. So I have the second part. It's pretty much. I'll try to be faster.
Sure.
But this is a polymerase. Actually, I did it longer time. So so we work on this bacterial phage system. So this bacterial phage from T7. So it doesn't need any transcription factor. So it is a good model system. It can also do things very fast. So experimentally, people usually use this to synthesizing something. Okay, so we choose this system because it's simple. It has multiple crystal structure at that time. So for this transcription machine, so you see actually, as we talk with Zen, so this idea of co-transcriptional translation can also happen. So you have a DNA generated, there will be positive reactive which I'll briefly mention in the end. So you see that there is a context, but right now we start with just atomically just look at its engine. So we think it's a mechanochemical and informational engine. Okay, I'll explain a bit here. So basically schematically, so this is a downstream DNA, upstream. So nucleotide taken to this to the active site added here. So basically you are catalyzing this polymerization reaction. So each step you add one nucleotide to this and throw a pyrophosphate product. After that, then you do the translocation, right? So right now it's still occupied by this newly added nucleotide at active site. Next, it moved downstream, and then this site will be weakened. So it welcomes the next nucleotide to come. So, but this part is mainly suggested with Brownian motion. We actually there are other proposals saying this could be some like power stroke or something, but we want to check so energetic of this. So the conclusion is Brownian ratchet, but it takes quite a lot of computation effort to confirm that. So this I put together. So this is a product release. This is a translocation. So both parts we did with just MD simulation extensively constructing Markov state model. So the conclusion for product release is it's needs a kind of activation. It doesn't do kind of energy drop. So it needs thermal fluctuation to have the product throw out. So for this translocation, so this is starting structure, this is end structure. Although they have different connection, population-wise, they are in the same order of magnitude. So that means they are actually the Brownian motion because initial and end are pretty much the same free energy. Okay, so this actually give you this idea of the ratchet, but right now it is Brownian motion. Basically, polymerase in this stage, they do not have preference. They can move forward and backward. Even Brownian motion, they can translate go forward and backward. But how does that happen? I mean, after this slide, okay, I add one more slide. So this is, we put the Markov state model thing together as a, I think you're just, okay, so they save time to play. So this nucleotide, the downstream will move to upstream. It's a lot of fluctuation. Okay, so the story, make the story faster. So this part we show Brownian translocation. It's like the system is kind of so sensitive fluctuation, and though later on they are kind of waiting to see which kind of nucleotide come. So the bias forward and or selection will happen in the next step to recruit the nucleotide. Okay, so this, I don't know if there's some, I think it is, okay, movie, movie I'm done, okay, play with you afterwards. Okay, so it's here I show some crystal structure. This is a cognate nucleotide. So this is a gating residue. So if this is a right nucleotide, soon it will form base pairing with a template. And then the gating nucleotide is set aside. And if you come with a non-nucleotide, even the deoxide, just one, remove one oxygen, then you see the template do not seem like it, and then the gating residue take over, right? The tendency is also obvious when you see GTP, which is non-cognate. It comes here, template goes away, the gating grabbed it. So these two are not that extreme, but if you calculate the interaction, you'll see that this gating residue have strong interaction. The template with this do not. So who tell is a cognate or non-cognate? It is a template. And by the reaction of the template, the polymerase then can tell, okay, the template doesn't like you, then I take you. So drag you there to maybe dissociate. So this is the initial screening. Actually, so yeah, so this is initial screening. And then actually, before between initial screening and the chemical reaction, there is also a pre-chemical state, which is insertion, or you can call it induced fit or something. It's basically to have your the initial bond nucleotide to further insert into active site to arrange better with the active site to prepare for chemical catalysis. Yeah. Okay, so to summarize this ratchet motion, initial Brownian motion, no bias. When you have the nucleotide come, it will bias forward, but only if they are good nucleotides. If the bad one, they can go back. So this is actually taken from the Feynman's ratchet, but it's different. This is a chemical free energy supported, not the temperature supported. By the way, for this T7RNA polymerase, it doesn't do proofreading, only do nucleotide selection. And actually this pre-chemistry step is a major checkpoint. So they also do screening, but then they do this kind of induced fit or pre-chemistry step. So when you, please.
Sorry, so for RDRP, for example, for coronavirus, once you have nucleotide incorporated into the RNA, you move 2 or 3 positions and then move back through backtrack.
Backtracking likely happen if you have wrong nucleotide happen. So this is also for COV2 system. So they actually, this COV2 system, they have proofreading. So it is related actually probably to this COV2, SARS-CoV-2 have a very large genome in the virus system. So they need some mechanism to do better. Usually virus do not do very well in Fidelity because they want to do mutation, they want to survive, right? But I think CoV2 doing better. That proofreading is usually related, not absolutely, but related to the backtracking is also usually related to proofreading.
So okay, the question is, since it can go past this initial attachment of adding a nucleotide and moving on.
It always, so take it always as the probabilistic. So even I say some nucleotide is rejected in the beginning initial, but there will always be some nucleotide leaks through. So it's not absolutely, so you get to this point, then you are all gone. You always have a chance to go through, but the chance could be very small. Right, so you want to calculate that actually. So this is exactly I brought this point. So then for each cycle, for example, for if you have cognate nucleotides, this is a pre-translocation, post-translocation, and pre-insertion, insertion product. So this is one cycle. For the wrong nucleotide, you also have a cycle here. And the wrong nucleotide is the backward transition usually faster. And the forward for the correct cognate one, it's usually the forward rate happens faster. So each step you can define some kind of selection energetic. It depends on the difference of either backward rate or forward rate. So because this cycle we cannot do it in empty simulation, is long, and so we do it with chemical master equation. So these some of this energy you can measure from empty simulation binding, initial binding energy, insertion thing, then you can put them together. So one step we didn't do is chemicals. You should bring principle doing QMM, right? But the thing is, if you know the error rate experimentally, so you can, first you can calculate in the steady state, the error rate, because you know both cycle, and then you can fit with the experiments, and then you can deduce back what's energy selection in the catalytic state. Like actually every kinetic state, there is a chance to do selectivity. So usually people do not measure that. I think it's taken knowledge-wise, it's difficult. So even experimental is usually mechanics maintained, but some people can do it very well. It's not easy. So then I switch gear to the SARS-CoV-2 system. We actually did also check this pre-chemistry, the selectivity. So this doesn't have structure and the machinery. This is polymerase. This is proofreading exonucleus, and this is a helicase. So it is the machinery collective work for replicating this SARS-CoV-2 for virus. And for the remdesivir, so this is a drug, right? So for normal nucleotides, they can do normal extension. So this remdesivir will get will will just escape the nucleotide selective control and add it. After that, it will terminate. So to do that, you have to escape the first the nucleotide selectivity, second the proofreading. Proofreading part related to maybe translocation, backtracking, which is more complicated. Some people try that little bit, already have publication, but to fully do that is difficult because you also need the helicase. And I think D Shaw with some collaboration, actually is those experimental people collaborate with D Shaw, they actually see some backtracking with this polymerase and nucleus together. So it will be very, polymerase itself already big. Then you have to have giant thing, but but as technical with strong, you may also want to take the challenge, right? Okay, so then what we did recently, published recently, is just we checked the selectivity of this remdesivir in binding and insertion into the active sites. So first thing we noticed that remdesivir actually form base stacking instead of base pairing. It formed base stacking initially, but later on it can form base pairing when they stably inserted. So this is a template. So this is remdesivir is sort of from kind of stacking, but there is some chance when we actually did the umbrella sampling. So you add force somehow, but in some configurations there can be some arginine interaction with phosphate, which actually seems to hinder the insertion of that. So we actually find the thermal fluctuation helps. So this is open to close. We put this as reaction coordinate. So along this way, we did umbrella sampling. Yeah, I guess I keep. So for T7 RNA polymerase, we actually also used this umbrella sampling to calculate the insertion energetic. So here is that the basic thing. Let me just be faster. So with when you have this in umbrella sampling, when you also control template, the barrier is high because there is some kind of interaction of arginine with the phosphate. But if you just allow the thermal fluctuation without enforcing on the template, you can see barrier is lower. So actually this is, again, the message is saying template is very sensitive to the thing coming. Okay, so in summary, for comparison of RTP insertion or ATP. ATP is the substrate, right? The two are very similar to each other. So initially they form stacking, and with fluctuation, you leave this sufficient in fluctuation of the template. So they can insert with energetic barrier even lower than ATP. And so this is pretty much what we get recently. Okay, so this part is by yes, rapid founding. Then let me quickly finish the supercoiling part. So for RNA polymerase, the DNA is not just passive. So the downstream will generate positive supercoiling, upstream will have a negative supercoiling. So this is a proposal, but then experimentally, they also detect that positive and negative in the two-way. And then there is a Sunny-Schiff-Map. They also find if they have this kind of gyrase, which can cut on the DNA. So during transcription, elongation, the positive supercoiling will accumulate and they will hinder the transcription. And if you put the gyrase there, they won't. So this hindering of this suggests giving the burst of the transcription in bacterial. So upstream is not quite an issue because they suggest that there are more polymerase there to do the job. So gyrase is just pretty rare. So there's their proposal. So we want to understand that by actually did the quantitative modeling. So because we do not know much physics there, we just do mathematical modeling at that time. So the purpose is that actually people try to suggest the two-state model, gyrase bound or unbound for that thing. But this process happened actually to several seconds several minutes. So we're thinking if you just take one step, maybe it's not good enough. So maybe we should model the accumulation step by step. Each polymerase, they transcribe, build some positive supercoating, and next build more. So we are thinking about many polymerases work together. So they have multiple steps. So then we can build kinetic cycle for single RNA polymerase or multiple RNA polymerase. Here we use some assumption. We suppose that supercoiling would be go together with transcription. The more you transcribe, you get more thing, and we think it's linear. We assume it's linearly. Okay, but this is assumption. At that time, we do not know this physics. So, but then later on, we kind of have a postdoc trying to do physical modeling of the supercoiling dynamics during transcription elongation, but he found it very difficult. So we just focus on supercoiling. Okay, so the thing is, he look at either constant kind of velocity of manipulating the end of the DNA, or just to constant talk to see how DNA react. So after quite a lot of development, so actually reading literature is pretty difficult, actually, this project. So we find we can actually have the kind of fast dynamic and slow dynamics. Fast dynamics is if you trace the DNA end, you see torque transport. And by writing down this energetic and diffusion equation, we can get the time scale of that. We can use actually polymer model to simulate the process. You see 0.1 millisecond to reach equilibrium. For that, we try to do the same speed, at least as a polymerase, although we cannot really simulate currently. So just try to do the same thing. But then if the most of the thing already is supercoiling, the thing would be different. Then we design a different, so this is another phase. It's called platonemic. So this is pretty much stretch phase. So we're dealing with two separate situations. The thing is here at the boundary of this, they should have phase this equilibrium, but inside the phase, the relaxation time is pretty fast. So our right now we just look at 100 to 1,000 base pair distance, and then we see that fast dynamic happen within individual phase, but the slow thing is we suppose it's at the boundary. So now it is this two-phase dynamics. So with this, we coarse-grain the DNA into one is this P phase, the other is this blue, this stretch phase. So just two phase, and then either we run polymer model or we run two-phase dynamic. We can always kind of reproduce this polymer. So our two-phase model, even coarse-grain can reproduce a polymer model. And first we kind of show. Actually, this process also happened. So supercoiling accumulation is comparatively fast. It happened within 1 second. So because if you accumulate it, it's several seconds. So it's a fast process. So you do not have to worry that the DNA supercoiling accumulate more and more long equilibrium they go to. So this is the one thing. The other thing is he can reproduce some diffusion, nucleation, hopping dynamics as measured experimentally by this two-phase model. So basically, this postdoc is physics background. He has very difficulty to thinking how to connect to biology. So this is also our issue. So when we do very physical model, it's still hard to go back to find the implementation, but we're thinking, so how to possibly link this coarse-grained model to a further interesting implementation. So for this, I kind of put our current interest direction, what I want to do at my webpage in UC Irvine. So in the end, so I just want to mention that I'm so happy to come back, but unfortunately Klaus is not there anymore. So last time I meet Klaus is when we were in Riken 2015 at that time. Yeah, so you see after Riken, it's K computer. Now it's replaced by Fugaku. Then it's already history, right?
Yeah.
It's a history. At that time, so Zhang is here. We take a picture.
Yeah.
So Sujita and I think this is Taji here. They are the host there. The host. These two my colleagues and. Yi San was from the show actually. So Klaus was not there. You know what he's doing? He was preparing his presentation. He said, I need separate time myself to pull down to prepare my presentation. Okay, so after that, Klaus, proposed to take a picture together. So Ahmad knows it's not easy for me, right? When I do presentation in the group, that was a difficult time. But that time, Klaus say, "Okay, you are doing good job."
Yeah,
I mean, I was very happy. I was very happy. So this was actually my last time with Klaus. Yeah. So okay, thanks for your attention.
Thank you very much. This was very nice, Jin. You have a lot of materials. Time for questions, I guess. I'm gonna actually start for your transcription part.
Okay.
So have you tried to do accelerated MD along a particular reaction coordinate?
You mean translocation?
For the translocation of the transcription factors.
A transcription factor.
The
Okay.
first part of your talk.
Oh, transcription factor.
Yeah.
We didn't, but I think it's.
If you want to do it, how would you? What is the reaction coordinate?
So exactly. So that is we are hesitating to do in the beginning.
Yeah, that's why you can't do it.
Yeah. So because sometimes you see they prefer rotation coupled, but if you change your concentration, they immediately react. But even in the normal ionic concentration, they have some chance to climb. So in that case, if you really still want to do, I think you can do both.
Yeah.
You can, once you pull it along the groove, then you can also measure the energetic. So as like in your kind of Markov state model or whatever model, you allow both paths to be possible. Then I think
then we have to also work with the center of mass of the protein and then its hydrogen bonds. To the DNA to be able to affect.
I think it's okay you dealing with central mass, but make sure the hydrogen bonding also follow. If they follow, then you are fine, right? If you see hydrogen bonding still there in the beginning, like my student tell me, okay, I think it moves, right? It doesn't really move, right? So they have to couple together. But because if you measure like the movement for several steps, it's natural you choose the protein central mass. Then I think there are other people doing that. They actually pull a pass, and then based on this pass, they do extensive sampling for Markov state model. I think it's also okay, as long as you are aware of they have a lot of stochastics.
Absolutely.
Yeah.
Any questions? Audience, also people on Zoom, if you have questions, I guess you can raise your hand.
I have one.
Go ahead, Dan. Please.
So, yeah, and I enjoyed your talk very much. Unfortunately, I had a meeting right before, so I couldn't make it up to Beckman fast enough. But I want to be sure I understand the very last part of your talk on the dynamics of the plectin of supercoiling.
Yes.
Dynamics. So your preliminary estimates, I mean, it's formation and relaxation. Because you don't have all the components in there. In my understanding, you think that goes on a millisecond rate?
Let me bring it back.
Like when you were solving that Langevin equation, so you're having it move along, but then of course it has to move and then eventually it has to relax, or are you just gathering more and more plectinines as you go along?
So actually in this slide, if we write Langevin equation, we just take the P phase and S phase. So it's not like because if you do polymer physics simulation, it's also take a long time to get that. So
Sure.
our logic is in the previous slide, we try to say, okay, we first check the torque propagation in the S phase and the P phase separately. We check the relaxation time. If you see that page, right? So this page, you'll see that 0.1 milliseconds they follow. So we rotate first, twist the DNA. First is stretch phase. The second, pure you already see plaque forms. We take it purely P phase, and then you see that when we change twist there, the response also reach to equilibrium about 0.1 milliseconds. Then we take this time scale below milliseconds as a fast dynamics. Okay, so this gives us the kind of intention. So we say, okay, something happened fast in this within two separate phases. Okay, then we assume that if we go above milliseconds, then these fast dynamics are kind of equilibrated within separate phases. Then upper in a milliseconds above time scale, we can treat them just like two phases because each of them are equilibrated within. Equilibrated means the torque propagation already relaxed. So that we can, with that logic. So next slide, next slide, we are trying to write down the Langevin equation just for just for boundary of that, the boundary of P and S. And then if we keep this scenario, that means later on, if we deal with 100 to 1000 base pair DNA and supercoiling, we can, if we concerned what happened in millisecond to second phase, or even to minutes, then we can just use P phase, S phase, and then do all the rest. We do not have to bother the polymer physics below.
Okay, so yeah, no, eventually it'd be nice to connect this with, you know, high C maps that they measure for contact interactions in DNA. But that's still pretty far away. But very, very nice. Then I had one other quick question from studying things like EFTU interacting with tRNA. A lot of those like lysine interactions with like a stem, like for an RNA, is we found more from electrostatics. When you say it's and so you were describing most of your motion through hydrogen bond type interactions. And did you ever calculate also just what portion of that is coming from from just electrostatic interactions? Or because of you use poly A, it's sort of washed out some way?
We do. In that, there's this summary figure. We just calculate. Sorry, just one thing. Let's see.
Yeah, I think it's more when you slowly moved over to sequence-dependent motion.
So here, the calculation diagram shown here is protein DNA electrostatic interaction. And also, if we do coarse-grained model, I think it's electrostatic interaction dominated. I think a lot of property actually can be motion can be described well by just protein DNA electrostatic.
Yeah.
But in the end, I think hydrogen bonding are very important. So especially you want to detect information. But why they are rate limiting each state? I think this stiffness have something to do with they detect some information there. So although because hydrogen bonding calculation, I think is more difficult in the sense, I mean, so but protein DNA electrostatic, we can usually gets interaction energy. So I usually do this interaction energy. So I think if you don't want to be very specific, I think the protein DNA electrostatic interaction usually play a very nice role. But down to the information level, I guess hydrogen bonding cannot be avoided. It is crucial.
Okay, all right. Well, thank you very much.
Okay.
Thank you, Zan. So actually, back to this sequence dependence, does this protein directly interact with the bases, or is it just all interaction with the back?
When in the specific finding, you can see they have very nice about 2 or 3 base. They have formed hydrogen bonding with the base.
With the base direct.
But not in other positions.
I
So
see,
this
I
is
see.
very nice. So
Yeah.
So this is really so.
Yeah.
How they gonna? So it's like they all form hydrogen bonding with the backbone.
Oh, yeah, I
And
see.
suddenly when they go to there,
Right,
they
right, I
kind
see.
of form with
Click kind of
Yeah,
into the
yeah,
bases.
exactly.
I
So
see,
we
I
want
see.
to understand that, but
I
it's
see, I
not,
see, I see.
I mean, we want to understand.
Yeah.
How.
Gotcha.
It's very
Gotcha.
important.
Okay, great.
Yeah.
Any questions from the audience? If not, please join me to thank Dr. Yu one more time. Great. Thank you. Very
There's
good.
a movie as a prompt.
Oh, you.
Sorry.
You can.
Yeah, I think it's confused with this change of the slide to the